Hi everyone. I can remember saying I was going to do a self-study for MCAT and post. So today will be my first self-study that I'll be posting, and it will be on the cell theory. Please let's say a quick prayer. And dear God in heaven, I pray that you grant us understanding even as we study. In Jesus' name I pray. Amen. So first, I would like us to look at the four tenets of the cell theory. One is all living things are made up of cells. The next is the cell is a basic functional unit of life. The next is the next is all cells come from pre-existing cells, and then you have the last being the genetic material of the cell is the DNA, okay, and it's passed down from the parent cell to the daughter cell. I hope we got the four right. You could okay. All living things are made up of cell. The cell is the basic functional unit of life. You have all cells coming from pre-existing cells, and you have the genetic material of the cell being the DNA, which is passed from the parent to the daughter cell. So a cell can either be a prokaryote or a eukaryote. And okay, the three domains of life are eukarya, which is a eukaryote, and then you have bacteria and archaea. So bacteria, archaea are prokaryotes. What's the difference between prokaryotes and eukaryotes? So prokaryotes do not have a nucleus, they don't have membrane-bound organelles, and their DNA is a single circular DNA, while you have the eukaryotes having a nucleus, having membrane-bound organelles, and you have the DNA being arranged into chromosomes. Okay, so I'd like us to look at the membrane-bound organelles of the eukaryotes. Remember, prokaryotes do not have membrane-bound organelles. So let's look at the membrane-bound organelles of the eukaryotes. So I'd like to start with the nucleus. So some refer to the nucleus as the control center of the cell. Well, you could say that because it contains the DNA, yeah? And then you have this DNA being organized into chromosomes. Okay, basically the DNA is being wrapped by histone proteins and, yeah, condensed into chromosomes. We will see that later in mitosis, where you have the chromatin being condensed into chromosomes, so replication is being done well, you get. Yeah. So and then you have, what again about the nucleus should we know? So the nucleus is being wrapped by the nuclear membrane, and the nuclear membrane is a double membrane, and it has nuclear pores. You have material being exchanged between the nucleus and the cytosol, and the cytosol and the nucleus. So remember, you have the nucleus being wrapped with a nuclear membrane or nuclear envelope, and it has nuclear pores that allows two-way exchange of materials. And then what again should you know about the nucleus? Okay, yeah, transcription occurs in the nucleus, and we'll see that later. So the next thing again, I'll go to I like to go to let's pick up the endoplasmic reticulum. So endoplasmic reticulum is continuous with the nuclear membrane or the nuclear envelope, and you see it is basically interconnected, like you see them, and then the endoplasmic reticulum could be rough or could be smooth. So the rough ones basically are being studded with ribosomes. You see the dot dot dots. You know, biology students, if you've done that before, and then you draw the intercalated, sacs and everything, the endoplasmic reticulum, and those where you put those dot dot dots, those are the rough endoplasmic reticulum, and they are responsible for translation of proteins. Yeah, basically proteins that are destined for secretion. Yeah, and then you have the smooth ones, the smooth endoplasmic reticulum. Basically, they are involved in lipid synthesis, and they are involved in detoxification. Okay, and then let's move on to lysosome. So the lysosome basically contains hydrolytic enzymes, and they break down cellular waste products. They break down materials that are being endosynthesized, and yeah, basically, and then if hydrolytic enzymes escape from the lysosome, the pathway just escapes, it can cause programmed cell death, which we call autolysis. Yeah, and yeah, and then so this autolysis will just lead to programmed cell death, as I said earlier, which is the cell is just dead. So you can call it apoptosis. I just mind my pronunciation. So next, I would like to take the Golgi apparatus. So the Golgi apparatus, basically they take care of modification. What they do, they modify the vesicles, the products from the endoplasmic reticulum. Let's say you have a detoxification occurs from the endoplasmic reticulum, or you have a protein coming in. So what they do, they is either carboxylate or phosphorylate. So basically they just modify, and then you could see them. They could also put specific signal sequences that would take those particular vesicles to the right cell location. Okay, so that's what the Golgi apparatus does. And then the next thing I'd like to look at is the peroxisomes. Yeah, so what they do is that they are involved in lipid synthesis and the pentose phosphate pathway. And but then basically what they do is that they break down very long chain fatty acids via beta oxidation. Okay, so and then one thing about the peroxisomes is that it contains hydrogen peroxide. Okay, and yeah, so hydrogen peroxide is needed for the peroxisome to function. And then the next membrane bound organelle. Okay, wait, we didn't talk about the nucleolus. So the nucleolus is where the synthesis of ribosomal RNA takes place, and you have the nucleolus as that dark spot within the nucleus. Okay, so when you look at the a nucleus of a cell, and then you notice a darker portion, that's the nucleolus, and then that's where ribosomal RNA synthesis occurs. Push. All right. So I hope that wasn't fast for you. So the next is cytoskeleton. So the cytoskeleton is very essential for the structure of the cell, yeah, for maintaining the shape of the cell, the structure of the cell. And then you have three types of cytoskeleton. You have the microfilament, you have the intermediate filaments, and you have the microtubules. So intermediate filaments are peculiar, like they're depends on the kind of cell. You have keratin as an example of an intermediate filament. You have lamines, you have desmin, vimentin, and then intermediate filaments, they ensure cell-to-cell adhesion. Me as a cell to you as a cell adhesion. That would be an intermediate filament. Intermediate, like between me and you, Anyway, so and then they also take care of the overall function of the cell. And then the next thing would be so let's take the microtubule. So the microtubule, you can see it in the cilia, you can see it in flagella, and the microtubule, what it does is okay, so motor proteins like dynein, motor proteins like kinin, they follow the microtubule, like creates a pathway for these motor proteins to transfer their materials, transfer their vesicles, their material vesicles, And then, okay, so one thing I'd like you to know is that the cilia and flagella of eukaryotes have a very beautiful structure. Well, those in prokaryotes are also very beautiful. I shouldn't be like that. Maybe because I'm a eukaryote, I want to, Anyway, so the structure of a eukaryote cilia and flagella have 9 pairs of ring. Remember, 9 pairs, that means there are 18, yeah? So 9 pairs, so like they are like 2, 2, 2, 2, 2, 2, 2 in 9 places, yeah? And then 1, 2, that's a pair in the middle, a pair, that's 2 in the middle, and 9 pairs around. So you have 18 if you're counting 1, 1, 1, and 2 in the middle. So 1 pair and 18 around, or you can say a pair and 9 pairs. So basically, you can refer to it as 9 plus 2 structure, yeah? While the cilia and flagella of this flagella of a prokaryote is arranged. You see the structure being a filament, a basal body, and a hook. So what next should we do? So I've not spoken about the microfilament. So microfilament, when you think of microfilament, just associate it with actin. And when you talk about actin, what comes next to mind? You have muscle contraction, where it works with the myosin to ensure that your muscles, And then the next thing again about microfilaments is that it ensures the structure, support, the cell and stuff like that. And you can see it in cytokinesis, like when the cell is about to break apart, when during mitosis or meiosis as well, when it forms a cleavage furrow. So at that cleavage furrow, you see microfilaments there, okay? And then next thing I'd like to look at, you have epithelial tissues, you have Okay, so basically there are four types of tissues in the body. You have epithelial tissue, you have nervous tissue, you have connective tissue, and you have the muscular tissue. So I'd like to look at the epithelial tissue and what tissue again should we look at today? Epithelial and connective tissue. So epithelial tissue, you can see your skin is an epithelial tissue, yeah, covering the body, and it also covers it covers cavities in the body, okay. And yeah, you see so you have the epithelial tissue being divided into basically you can divide it based on the number of cells or based on the shape of the cell, and based on the number of cells you have either is simple as just a layer, or you could have multiple layers, which is stratified, or you could see even pseudo-stratified. Basically, pseudo-stratified is actually a single layer, but then it appears as though it's multiple because of the difference in the cell heights, okay. But then don't be deceived, it's a single layer, but then it just appears as though it's this multiple layer because of the difference in the height of the cells, okay. And then you have based on shape, you have squamous, which is flat, and you have your cuboidal, my pronunciation, you have cuboidal from the name cube shaped, and you have what columnar from the name column shaped. So you can have squamous, simple stratified, you can have cuboidal, simple stratified, or you can have columnar, simple stratified epithelial cells. Next, I would like to look at, you have connective tissue. From the name connective tissue, you'll be expecting support, yeah. So connective tissue provides support. I love connective tissue because then you see your blood is a connective tissue, you see your adipose tissue is a connective tissue, yeah. And you see what again, your cartilage, your tendons, yeah, your bone, yeah, those are all connective tissue. So next I'd like to look at, you have yeah, so I'd like to look at bacteria. Remember, bacteria is a prokaryote. Under prokaryotes we saw sorry, under prokaryotes we saw bacteria and archaea, okay. So bacteria is a prokaryote, so you would expect it to have a single circular DNA, okay. You expect it to not have a nucleus, you expect it to not have what again, to not have membrane bound organelles, yeah. Okay, so what again should I say? So when you look at bacteria, you would see that, okay, they have, some bacteria could have 1, 2, or even more flagella, yeah, and yeah, covered in cilia, and yeah, so bacteria could be an obligate anaerobe. When it's an obligate anaerobe, it means that it cannot survive where there is oxygen. Is obligatory, like obligate anaerobe. So anaerobe means that it doesn't need oxygen for its metabolism. It could use chemosynthesis, yeah, so chemicals, other sources, inorganic, yeah. So basically, I hope I didn't get that mixed up. Anyway, so obligate anaerobe means that it does not need oxygen to metabolize, and it cannot survive where there is oxygen. And then obligate aerobe will mean that it cannot survive where there is no oxygen, okay. And then you have facultative anaerobe, in which, okay, I have oxygen, I'm going to use oxygen. If there's no oxygen, I'm going to use other means, chemosynthesis, I'm fine either ways. And then you have your sorry about that, you have your aerotolerant anaerobe. So aerotolerant anaerobe means that I'm an anaerobe, I will perform metabolism where there is no oxygen. However, if there is oxygen, I can tolerate oxygen. I'm not an obligate anaerobe that I cannot, but then I'm aerotolerant, I can tolerate oxygen, yeah, but then I will not use oxygen to perform metabolism, okay. So I hope I didn't confuse you with that. You could go back to like get understanding, like better understand that. So and then okay, so you have bacteria being classified based on shape as well. You have cocci, which is round shaped, you have bacilli, which is rod shaped, and you have your spirili, which is spiral shaped, spirili. This anyway. So the last thing I like to look at would be cell wall, the cell wall of the bacteria. So you can know the type of bacteria by its cell wall, whether it's gram positive or gram negative based on the cell wall. So what you do is that you would crystal violet stain that thing. You stain it with crystal violet, and then you will counter stain it with safranin. Okay, so when it turns purple, when you do that, you know it's gram positive. And you know what gram positive means? It means that it has a thick layer of peptidoglycan. It has a thick layer, and then what again? And it also means that sorry, I'm trying to. Yeah, it has the cell wall is thick. It means when it turns purple, it means the cell wall is thick and it's composed of peptidoglycan. Okay, and then you have that. It would also have what's the name? It will have lipoticoic acid. And you know, funny thing is that in terms of antibiotics, when a bacteria has been attacked by an antibiotic, so gram positive means that it has a thick layer of peptidoglycan, okay, and it has lipoticoic acid. So it's the lipoticoic acid that would actually, like, when the antibiotic resistance come at it, is the lipoticoic acid that will like react or something, like you know, the word has flown. Anyway, next is the gram negative. So gram negative will have a thin layer of cell wall, so thin layer of peptidoglycan, and it will have an outer membrane that is made up of phospholipids and lipopolysaccharides. So in gram negative is the lipopolysaccharide that is that the antibiotic will like attack, or like when it comes at the gram negative is lipopolysaccharide, when it comes at the gram positive is lipoticoic acid. Yeah. So what next? So I said that was going to be the last thing, but then I forgot something very important. So let's see. I would like to I'm so sorry, I hope this is not boring anybody right now. So next is genetic recombination. Yeah, so I would like to look at bacteria genetic recombination. So you have I'll look at transformation, transduction, and conjugation. So for transformation, this is when the bacteria, yeah, it will pick up exogenous material. That means it will pick up genetic material. It will pick up genetic materials that are outside the cell, exogenous genetic material. Okay, I'm a bacteria, for instance, and then I pick up exogenous material, exogenous genetic material. There are genetic materials outside me, and then I pick it up as a bacteria. Yeah, so that is transformation basically. So and then you know the funny thing is, as I'm picking it up into my genome. I shouldn't leave that out. So I pick up an outside genetic material and put it into my genome. That's transformation. Transduction is when a bacteriophage is involved, and bacteriophages are viruses that infect bacteria. Okay, so you have a bacteriophage, like, okay, you have a virus now called a bacteriophage which infects bacteria. So it will infect this bacteria, and then that's being the meaning of like it infecting means that it's injecting its genetic material. Yeah, so this virus will inject its genetic material into the bacteria, and then instead of it to carry back maybe its genome, it will carry, let's say, the bacteria's genome. You know, it was a virus, it infected its virus genome into the bacteria, and then it will now mistakenly carry the bacteria's own genome and go and infect that bacteria's own genome that it carried into another bacteria's genome. So you see that a bacteriophage is a virus that infects a bacteria, okay, and it carries genetic material from one bacteria to another bacteria. I shouldn't be actually saying this bacteria, it should be bacterium, because bacteria is plenty and bacterium is one. So I hope I didn't confuse you. Sorry. So next would be conjugation. So conjugation is so let's say you have a bacterium and another bacterium. They will form a conjugation bridge that will transfer genetic material from one bacterium to another. So bacterium 1, bacterium 2, and they have a conjugation bridge that is transferring genetic material. So the one that transfers to the other, the one that is transferring is the genetic male, yeah, and you could see well, it's usually a plasmid that is being transferred. So the plasmid from the male will be transferred to the female, yeah. So you have your okay, the plasmid, let's say the plasmid is a sex factor. You have F plus being transferred to F minus. Okay, so F plus is the male donor, F minus is the female recipient. Okay, and then is the male that forms that conjugation bridge. It's from it. It has a sex pilus, pili, plenty, pilus. So it will form it, and it's because of that sex factor, which is the plasmid that it has. And you know, a plasmid is an exogenous genetic material. Well, no, it's not exogenous, it's extra chromosomal because the plasmid is outside the chromosome. The plasmid is outside the chromosome. Do not forget that, okay? The plasmid is extra chromosomal. I hope I'm not confusing you, please. Then, what again? So, that's most of what I got from my first study, and I would like to look at viruses, but then okay, I would look at viruses after this out. Yeah, so I hope you liked this study and you were able to learn a lot as well. Thank you very much. See ya.